an inhibition of reverse transcriptase experiment. In this case, for the same experiment as before, we used color 3 plus minus RT. So they did not overexpress line 1, we infected, and then we did again the tagmentation procedure to look at integration. And here we look at just the DNA sequences. In the non-AZT experiments, we found a certain signal of DNA in the DNA, but much less in the RT experiment. So you would argue AZT, the RT inhibitor, reduces integrated SARS-CoV-2 sequences. So let's come to line 1. Line 1 encodes 2 ORFs, one ORF1, which is a RNA binding protein, and ORF2, which encodes the RT and the endonuclease. And so they really have a big part of the human genome, 17%, and about 100 active copies are in the human genome. They're active in early embryos and in brain tissues. So what affects line 1 expression? They're normally expressed at a very low level. And it's known aging does. So when you look at, and this comes from a recent paper, they find in aged fibroblasts, there's high upregulation of line 1 expression. Similar in cancer. In cancer, there is much upregulation of different flavors of line 1. And that can lead to activation of oncogenes. So we looked also, can line 1 be activated by SARS-CoV-2 infection? We looked here just at published data where people compare infected with non-infected cell lines or in patients, and line 1 indeed is in the infected ones significantly upregulated. And this will be different. There are different elements again of line 1 which are upregulated in these in culture infected cells. And this is an experiment we did in another cell line, and we find a 2-fold upregulation. So viral infection, stress, induces line 1 expression. We asked the question, can cells which are exposed to cytokines, like in patients, cytokine storm, can they activate line 1? And here we expose cells with a microglia conditioned medium, so we're just with cytokines. And you can see we find now by looking for ORF1 expression, we find line 1 ORF1 expressed after exposure of these cells to this conditioned medium, and this gives you about the quantification in different experiments. So cellular stress, viral infection, and cytokine exposure can induce line 1 expression. Let's keep this in mind. So retrotransposon line 1 repression, if I summarize this, can be by genome-wide, that's really by quite a lot of different groups, line 1 expression by regardless of virus. So many different viruses, in also probably DNA viruses, can induce line 1. Cytokine exposure of cells, I showed you the data, and aging cancer. They all can lead to de-repression of line 1 expression. So if I summarize my first part of this. So SARS-CoV-2 subgenomic RNA can be retro-integrated in the genome of cultured cells, and clearly majority by a line 1 mediated target site primed reverse transcription mechanism. The fraction of cells with integrated copies is low, and we can only estimate this. It's probably 1 in 1,000 to 1 in 10,000 infected cells in a culture, which is very similar to what Zinkernagel observed in the LCM viewers. So it's low. And as I said, line 1 expression is induced by cellular stress. So the key question is, can integrated SARS-CoV-2 sequences be expressed? I think that's a key question. Can it be expressed in cultured cells, or can it be expressed in patients? So let me come to this. And this brings me back now to experiments we were forced to publish in December in a preprint on bioRxives, which I regret deeply that I ever did that, but we were forced. So let me go through these data. So we looked at, to confirm, we looked at in published data, were there any chimeric reads between virus and cellular RNA by using, just analyzing these data. And we did find chimeric reads in many of these cells, and particularly in some patient rates, very high. So it's plotted the total fraction of mappable reads against the chimeric ones. And the conclusion was there was a fraction chimeric reads between 0.04 and 0.14%, but up to a maximum of 64%. I will come to this back in some patient-derived lung lavage. So keep this in mind. And this just gives you which sequence of the virus were found in these mostly from the 3' end, the nuclear capsid, but all other were found, also the spike and also the other proteins. So this was consistent with integrated sequences being expressed. That's what we put in the paper, and we got really beaten up for that. I think justifiable, because there was a problem. And the problem came out in this paper in February, where they looked at this and they concluded that the error prone due to random template switching during reverse transcription of RNA to cDNA. So the RT jumps between templates. And they estimate that 1% of the RNA sequence reads are artifactual chimeric. So our conclusion based on these chimerics were absolutely non-tenable. It was an artifact. So I have to be honest about this. And as I said, I regretted deeply that we ever put this in this preprint. So we were under a big pressure to solve that. And so we thought about different criteria for expression of integrated sequence. Are they? And let me go through the logic here. So if this would be the normal infected cell having plus strand RNA, but if this RNA would integrate by retrotransposition into the genome, there would be 50% that would be the same orientation as the human gene they integrate to, and 50% the opposite. There should be no preference, right? So does it happen? And when we look at the DNA in our sequences from the nanopore sequencing, indeed, 50% were in the same orientation as a gene they integrated into, and 50% the opposite. So the opposite means the minus strand of viral RNA was fused to the plus cellular RNA. So the prediction was, that's important, 50% of chimeric reads would be from integrated copies should contain negative viral strands, and 50% positive. So key was now how many viral minus strands are in an acutely infected cell. So we looked at this in our own data and published data, and it's very low. It's between 0.01 and 0.01% of all viral reads are negative strand. So very little negative viral strand template is made in acutely infected cells. So the acutely infected cells, the ratio of minus to plus strands is less than 1 in 1,000, which really eliminates basically any artifactual chimeric generation doing library preparation. So keeping this in mind, now we look at patient samples. Patient samples from published data coming from the lung, for example. And when we look at this from this published data, we find now in these patients here, we find many of those contain negative viral strands, orders of magnitude higher than acutely infected cells. Some contain 50%. 50% basically means, if you follow our logic, 100% of the viral reads come from integrated sequences, and some others were less. Are these negative strand viral strands seen in the chimeric reads? For those, we could look at them. There were not that many we could detect because of the length of the reads. Yes, some of those had 50% of the viral component in the chimeric reads were negative, and some others had less. So the conclusion was mostly negative. That's important. From the clinical data, from these data, there was no evidence for viral replication by immunohistochemistry. So this comes from again, virus-free cells. And they made so much negative viral strand RNA. In another set of data was single cell sequencing. When we summarize them, all of these patients had really gained orders of magnitude higher negative strands, and we look at their single data, depending what your threshold is, with 10 viral reads per cell or 5, we find in some cells very high frequency, again up to 50% in some of negative strand viral RNA in these samples. So in some patient-derived tissues, most, if not all, viral transcripts are derived from integrated genomic SARS-CoV-2 copies. Let me get a key conclusion in this. This, I think, is indirect evidence for integration of SARS-CoV-2 in the genome of patient cells. I don't have any other way to explain that. And I want to just right away say, to directly see viral integration into the DNA of patients, I think is beyond all our methods we have. It's too rare. If it's 1 in 100,000 or 1 in 10,000 cells, no way we can detect this. But it's indirect because of it's amplified by RNA expression. So let me summarize in my second summary. So SARS-CoV-2 sequence integrated into the genome can be transcribed in some patient-derived tissues. Most, if not all, viral transcripts may originate from integrants in those, in the absence of viral application. And this could certainly explain why many patients are PCR positive long after they one cannot detect a detectable virus anymore. Okay. So I must say our paper met a lot of criticism and I think sometimes open hostility. I was very surprised. I'll give you one example. So this is a paper which came out last week. And what they've sequenced and what they conclude is they found no evidence for SARS-CoV-2 integration. And so it's very unlikely that our data are correct. So let me base what they did. They did nanopore sequencing of infected 293 cells, something we also did. They had a positive control. And the positive control was HBV insertion. They found one HBV insertion in liver. Now, HBV is known to integrate by a totally different mechanism. So finding one is not a terribly convincing positive control. Let's compare this with our data. Our data were based on nanopore sequence of infected cells that overexpressed line 1. I talked about this because we wanted to find as many integrations as possible. But for non-overexpressing cells, which correlates to what they did, we did not use nanopore sequence. We thought it was much too insensitive. We used this target enrichment fragmentation sequence, which is much more sensitive. And we found 10 integrations with, as I said, all had line 1 recognition sequence in the flank. So the data are totally relevant for our conclusions because they're using an insensitive method. But they make these very strong conclusions. And then they made another curious statement. And they saw the unusual 28 kb internal deletion of SARS-CoV-2 sequence they have criticized. Well, as I said before, this is not a 28 kb deletion. It's really the subgenomic RNA, which is missing 28 kb sequences. So it's really, they ignored and they didn't know about the virus replication cycle. Okay, but there are many more of those I could give you. So there are many open questions. So what is the prevalence of SARS-CoV-2 retrointegration patients? As I said before, it may be a very small fraction only, and that's based on the LCMV data and what I showed you. And I believe it's no way we can detect this directly in patients. We have to do it indirectly by looking at the RNA. But with more than 140 million humans infected, even a small fraction may be significant. So what are the consequences of viral integration? This is no speculation. I think the key thing is, do they express viral or neoantigens? And we do not know. If so, if they express a viral antigen, could this give you continuous protection? Or could it elicit an autoimmune response, such as in the late complications? Do other disease-causing RNA viruses, influenza, Zika, dengue, and so on and so forth, would they also retrointegrate? I believe any RNA is subject to this integration if line 1 is introduced. So what are the possible consequences for the course of disease? And this is all speculation, I do know. So be sure you take this with a grain of salt. So let me come back to the LCM virus from Zinkernagel. So this is a paper no more than 20 years ago, and they found in vitro and in vivo studies in mice, they found that reverse transcription, as I said, led to LCM viral RNA into complementary DNA, which could be found in the DNA. And they concluded, they speculate, which I think is interesting, which I want to quote. The ability of antigen-presenting cells to accumulate viral DNA genomes thus potentially represents a naturally produced form of DNA vaccine. Total speculation, but I thought it was interesting to bring this up here. So let me come to the autoimmunity issues. And as you know, and there are quite a lot of know, Guillain-Barré is seen as an autoimmune consequence after late stage after COVID infection in quite a lot of patients. And this is a few examples. And there are actually many more. The partial list of autoimmune conditions are Guillain-Barré, immune thrombocytopenia purpura, so the autoantibodies against platelets could be lethal, deep vein thrombosis, pulmonary embolism, stroke, disseminated intravascular coagulopathy, very often seen, very serious, multi-system inflammatory syndrome in children, the Kawasaki-like disease, type 1 diabetes, myasthenia gravis, Graves' disease, and the list can go on. This is really the big concern. Where does it come from? And I was interested to see this paper, I know a few weeks ago, where they looked at only few patients, only 7 patients, and they isolated cells, B cells, from the liquor of these patients, and asked, it produced monoclonal antibodies from these B cells. And what they found is, in 5 of the 7 patients, they found that the antibodies, monoclonal antibodies, reacted against neural tissue or against viral antigens. But interesting, one antibody reacted to both. One monoclonal reacted to both spike and neural tissues. Now, this, of course, could be certainly involved in the pathogenesis and how would this come. So I think that's an interesting issue, although it's really a very small study. So we come to another issue. What cells are really the ones which replicate the virus in the body after infection? And so which cell types are susceptible to infection, and if they're carrying integrated copies, could present potentially viral antigens long-term, and thus possibly evoke an immune response. And so we went ahead to look in ES cell-derived different cell types, just making whatever cell types we could make from ES cells or from iPS cells and infect them with the virus. So viral tropism infection of ES-derived cell types, of course, this requires work with infectious virus. In the BSL-3 facility, and Alexa Richards, she's the only one at MIT who's authorized to work there because she has the right training in the Reagan facility where she can work with infectious virus. So how do we detect SARS-CoV-2? 3 assays. One is a plug assay, so this virus plugs very well on its susceptible cells. PCR, to look at viral RNA, or immunofluorescence to look at viral proteins. So we used all of those. So this would be, for example, immunofluorescence for double-stranded RNA, which only do in cells which replicate the virus. You can see in these cells, which are CALO3 cells, which really replicate, well, you find cells are really expressing double-stranded RNA. PCR, this gives you an overview of all the different cell types we use. We find that astrocytes, sensory neurons, cortical neurons almost don't express at all. So this is not PCR. This is infectious virus, right? Infectious virus in the supernatant. Hepatocytes are this is a logarithmic scale, very well replicating cells. Smooth mother cells, we'll come back to those, very well replicating. Endothelial cells really are not infectable. Pericytes, cardiomyocytes, much less. Lung epithelial cells, either they were directly, primary cells were highly infectable and could produce virus. Then we come to liver. Liver, we made liver organoids, and they really produce virus very well for quite a lot of days, and there seems to be no pathology on the liver, so they don't die. And they really activate the immune response, but I don't want to go into this in detail now. Intestinal organoids, when you expose them to virus, they express a lot of virus, but then it goes down. And they die, these cells die. So that's probably what the reason is. We wanted really to look at the vascular disease. As I said before, this is really a serious complication of the infection. And so we wanted to look at those. And the key cells in the endothelial is a damage to the endothelial barrier by direct infection of endothelial cells. So these are the cell types we looked at, the pericytes, the endothelial cells, and the smooth muscle cells, which are all important for the barriers. And so virus infection of these cells, all made from embryonic stem cells in culture, and they were exposed to virus. And when we looked at them again, the plaque-forming units produced by their endothelial cells basically are not infectable. The virus titer goes down, it's probably just the input virus, which disappears over 2 days. But smooth muscle cells really replicate the virus. That's really the logarithmic scale. Mesenchymal cells do also replicate virus, less so, and pericytes do. So smooth muscle cells. When you look at those, this would be a non-infected smooth muscle cell, and this would be infected ones. So they clearly change their actin skeleton, and they change their shape, but they really produce double-stranded RNA. This is green, but they don't die. Endothelial cells, either mock or not, there's no evidence for double-stranded RNA, so they're really resistant, and also by this assay. We look at cortical neurons, and that's of course important because of the brain issues. So this would be a positive control, Japanese encephalitis virus, which we work with. So when you expose neurons, cortical neurons made from ES cells to virus, we find active virus replication as seen by the double-stranded RNA in green. And of course, it's also in the CALO3 cells, which is the lung cancer line, which we use to produce virus. But if you expose these neurons to SARS-CoV-2, there's no evidence for virus replication, but they can, of course, replicate in the CALO3 cells. So the conclusion is, really, this virus cannot infect neurons. They're lacking the receptor. So all these serious brain symptoms we see are probably indirect, probably a consequence of some immune-mediated damage. But we don't quite understand this. So to summarize all what these tropism experiments told us, CALO3 cells, 293 cells, all standard cells, they're very well, they produce a lot of virus. Neurons, really very little, if any. Astrocytes, very little. Liver cells are quite well in producing virus. Endothelial cells, absolutely not infectable in our experiments. Smooth muscle cells, as I said, very highly infectable, and they survive. Pericytes, less so, in mesenchymal cells also produced, and cardiomyocytes, we can detect virus replication, but less so. So my final summary, cell types differ in the ability to replicate the virus. There's clearly differential sensitivity to virus-induced cell death. SARS-CoV-2 does not infect endothelial cells, but can replicate robustly in smooth muscle cells. Now, whatever that means for the disease, of course, we do not know. That's all in culture experiments. And liver and lung can efficiently replicate virus, again in culture. So the questions which are there some cell types that can replicate a virus, but don't die, but survive long-term? And maybe candidates, maybe the smooth muscle cells, and maybe liver. We do not know. And could such cells present an antigen if they carry these integrated sequences, the absence of viral replication? Could they present the immune system for prolonged times? And I should say, what fraction of this would you need in cells? The fraction might be very low, very small. So I think the key thing will be to really understand, can these integrated sequences give rise to transcripts which can be translated? And what would be the nature of such a protein? Could it elicit an immune response? And we are totally ignorant about this, and it will be not an easy task to get to this. So at least there are many, many questions raised by what I told you, but I think it will be of quite some interest to learn more about this. Maybe we can understand more about the biology of this virus and maybe other RNA viruses. Okay, so let me come to the end. I really acknowledge the people who did this. Ligo Zhang and Alexia Richard were the key drivers on this project, and a lot of help from other people in my laboratory, because it was a lot of work to produce all the cells and many other of these. So they are all participated. I always have a really quite close collaboration with Rick Young at the Whitehead Institute. Inma Barasa in Whitehead Computing, very important for all what we talked about. Steve Hughes from the National Cancer Institute, who really had major intellectual input in what I said, and Nathan Islands for some inspiration. And with this, I'm happy to answer questions.
Thank you very much, Rudolf, for this really fascinating lecture and really for sharing this data, which clearly stirred up a lot of people in the community. I have a couple of questions to get started, and then hope that people will just either unmute themselves or put questions into the chat. So a first question that comes to mind for me is that it's clearly a rare event, the integration, and the expression is even rarer. But if it happens, what is the worst consequence? I started off by saying you explored the latency potential of this virus. So the question is, do you think there will ever be, or could there be an entire copy that gets integrated that could actually become activated again? Or if it's fragments, as it is what has been found, it's mostly fragments that get integrated, could there be even a worse scenario in this way, that different fragments get put together in a way that it constitutes a new, even worse virus that we would then have to deal with? And if I can add one more point to it, the fragments that were integrated but were lowly expressed, did you ever get a chance to look at the epigenetic marking of these fragments? Because that could, again, of course, influence the level of their expression.
So, yeah, I think these are all very interesting questions. So first of all, I think it's no way at 30 kb genomic DNA could integrate. No way. So what we find, the highest one we found was 5%, the whole nuclear capsid, right? 1,600 base pairs of 30 kb is the whole genome. And this totally reflects the prevalence of the nuclear capsid subgenomic RNA. It's just a matter of probability. That's the highest expressed RNA in the infected cell, and that can get with the highest probability integrated. It goes down for all the others. So I think there's just no way if you get this. So your question, can they be spliced together? So I think given the rarity of integration, which I try to really point out, it's rare. I think that's almost beyond any expectation that there could be in one cell accumulating several of those subgenomic RNAs which could splice together and get all the non-structural proteins, which are much less highly expressed, much less likely to integrate. So I think I would almost exclude that part. Yes, so that's, I think that was all what...
The epigenetic marking,
So that's really an interesting question. So we were worried about this. So given that we cannot look we can only look at these population of cells, we can't clone the cells, the cells, infected cells die. So we have really just to stop at the total using the sequencing data. But nanopore sequence allows you to look at methylation. Maybe you're familiar with this. I was totally unfamiliar with this. But in the very first experiments, we used that, and I can't give you the evidence for this, but you can look at the frequency of methylated versus non-methylated Cs from the nanopore with the software which I cannot explain. So we did the very first experiment, and indeed we found that we looked at these two integrants, which we had both copies. Were they in methylated, integrated? First of all, they were unmethylated in the few Cs we could see. And what was the surrounding? And in one of them, the surrounding couple of Cs we could see, they were methylated, and in the other one, they were not. So there is a possibility to look at this using this technology, right? But this is a very, I mean, it's a totally preliminary result. I just want to bring it up. Yes, one could look in principle at this, but it will be a tough call.
Okay. Well, thank you very much. We got a question from David H. Horowitz here. Very interesting talk. Have you studied the infectability of glial cells, which predominate in the human brain?
Yeah, so as I said, we looked at astrocytes and they were very poorly expressed. We looked at microglia, they were poorly expressed. So those are two we can routinely make in the lab. And so so if they get infected, it's very low. It's a very low frequency. We looked also at neurons. As I said, neurons, we didn't find any evidence, and that's also seen from clinical data in autopsy brains. There was no evidence for virus replication in neurons. We looked at peripheral neurons because of the smell, anosmia, and there was some replication of those, but again, quite low, but it's clearly more than cortical neurons. So the cell types vary in their susceptibility to virus by these in vitro assays.
Can I ask you another question, more political? You commented on the bioarchive issue, and I would be interested to know who actually forced you to publish in bioarchive, because I have never heard that. And also, wouldn't you say, having been forced and having published in bioarchive, it actually did highlight this switching thing. So it actually served its purpose, wouldn't you say?
Well, I regretted it deeply. So the journal we submitted this paper to said as the condition with submission, because it was COVID-2, it has to go on bioarchive. And I think it's totally unreasonable, and I was stupid enough to accept this. And I got burned by this. Because now it was a public, I thought we could better it out with the reviewers. I knew we were lacking some data. I didn't know about the artifact I talked about, but we were lacking integration data. That was the key, and I thought we would get it very fast. It was much less fast than I thought after we submitted the paper, because there was some pressure. So that was my mistake. It was, the journal forced us to do this. And now it was instead of a discussion with the reviewers, it was public and was not a good thing. So my policy would be, I would never do this again. If a journal insists on that, I just withdraw the paper, because I think that is absolutely, it doesn't serve anything to do this, to my opinion. There are other reasons why I would publish in bioarchives if a postdoc wants to get it on the job market. But I would never be forced by a journal to do this. I think it's totally unreasonable. And so we got burned by this. And you know, it's interesting. The conclusions of this preprint were exactly the same as what we published in the PNS paper, exactly the same, except they were not supported by credible evidence. And that's what I have to say, yes, mea culpa. That was my mistake. So we learned.
Yeah, no, I think that sounds very reasonable. We have a question from Mark Mansour. It's sort of an enhancer-like question, stimulating talk, thank you. Is there any evidence that integration can have regulatory roles such as enhancer function?
We have no evidence for this. I mean, the RNA which integrates, of course, doesn't have a promoter, right? So it's just the RNA. So it can be expressed only in really fusing, it integrates into an expressed gene, and we see that, of course. So then it gets expressed as a chimeric transcript. We have really no evidence that integration into an intron or into an intergenic region, which we find that they're expressed. We have just no evidence for that.
Okay, I don't see any further questions. Also, if people just want to ask questions rather than typing in, sometimes it's easier to just formulate it if you don't know how to put it into words. But if not, then we really would like to thank you very much. Oh, here we have another question from Lauren. Thank you very much for a stimulating talk. Could the effect of aging on line 1 aging account for the age-specific post-COVID symptoms?
Yeah, so I think that's exactly what I believe is I think consistent with the risk factors for for long-term bad symptoms after infection. So age clearly is the biggest risk factor, as we know. And there are certainly other conditions like obesity is known also to activate inflammation, activate line 1. So these are all risk factors that could really stimulate integration from a very low to maybe less low level. Could this be important for the maybe rare long-term effects? I think we only can speculate. We don't have the data for this. Cancer, the same thing. Cancer can be a risk factor for COVID long symptoms. They're overexpressed and induced clearly, as I mentioned, induce line 1. So I think line 1 expression is an important issue here. And line 1 expression, there was a recent paper, line 1 expression by stress, by virus integration, is thought to stimulate the immune system, the innate immune system. So there could be actually some function in that. So I think we are pretty ignorant about this, but all this, if you follow our logic, could stimulate the low frequency of COVID integration to less low frequency.
Thank you very much for that answer. I was wondering if there's a connection between this and also the more recent extra chromosomal DNAs that they're finding in cancer. It seems like there could be some interesting overlaps there.
So I don't know this at all. So if it was line 1, it will integrate, right? So this is the mechanism which line 1 itself uses, and as I showed you, the retroposition of trans of RNA. But so coming back to the VSV data, I mentioned this paper, 10 years ago. So what they found was in an acutely infected, VSV infected cell, human cell, you find DNA copies in the cytoplasm, right? As also Zinkernagel found. And they found it was fused to, in this case, to IAP elements in LCMV. And they also found it fused to some of these radiotransposons. So those would be, if they integrate, would be very different mechanism. It would be not retroposition. They would integrate like a plasmid integrates, presumably at double-strand break. And so Zinkernagel didn't find any target site duplication, and we find in 30% over integration. We didn't find a line 1 a recognition sequence. Of course, we don't know whether it's target site duplication. So it could be that these DNA copies in the cytoplasm could hang around for a while. We don't know. We have really no good evidence. We have zero evidence for this.
Again, if they get chromatinized, maybe they survive even longer then.
They could. I mean, it's a possibility that they hang around in the cytoplasm for a while as DNA copies. And would they be transcribed as DNA copies without having a clear-cut promoter? Maybe they have it from the virus. I don't know. I think these are pretty interesting questions which sort of come up now because people didn't think about this. I mean, I really think this is nothing special for coronavirus. Any virus, or probably any RNA, will be subject if line 1 is there for this type of integration. So we looked at other viruses, Zika virus, dengue virus, we did that. And we didn't find direct evidence. The reason might be that these are pretty toxic viruses. So you don't have much time to wait. So it could be below the detection limit, I don't know. So I assume other people may be more competent than we are will look at this. And I would be surprised if they're not subject to a retrointegration by this mechanism.
Yeah, you certainly started to stimulate the community to ask these questions and look into this in a little bit more detail. Right, I think I've no further questions I can see, and we have also run out of time. So let me thank Rudolf again for this wonderful lecture and close on a promise. Usually, under normal circumstances, we invite our distinguished guests to UCL for additional discussion and hospitality. And so what I like to do is follow this up with an invitation once the pandemic is over, and I hope you will be able to accept again and visit UCL
Sure.
in person.
I would love to come. Sure.
Thank you
Yes.
very much, Rudolf.
Well, thank you for listening. And yeah, goodbye.
Thank you. Bye.
Bye.